Recently, a number of us have been working on a technology training grant at Naugatuck Valley with the goal of trying to digitize many of the slides that are contained in the slide box collections in the anatomy and physiology laboratories. At this point, we have a good number of images that have been photographed, and the question now is, what are we going to do with them? Some of them we can just make available to our faculty, and they can do whatever they like with them. But we thought we'd try something a little bit different here. I'm not so much concerned about the presentation aspect of what I'm doing right now as the technology. I need some feedback as to whether or not this is coming across clearly in your home computer. I'm going to do a couple things right now and test the capability of this. And explaining as if we're sitting next to each other, looking down the eyepiece in a microscope together simultaneously. So let me point out this is a plant cell. First of all, it's a group of plant cells. This is plant tissue, and this is an onion root tip, particularly in the meristem region, which is an area of high cell division or a high mitotic index. And if you look over on the left-hand side, at the bottom left-hand part of this picture, you're going to see an area where the cells don't really have any kind of distinctive structure within their nuclei. These cells here are in interphase. Now what I'd like to do is find out if I can possibly maybe even type that in over here. So we're going to take our little cursor over here and go INTR. And that seems to work actually. So we'll put this over here. These are interphase cells. Okay, the interphase aspect of a cell's life cycle may or may not include at any given moment duplicated or unduplicated chromosomes. If you recall, interphase was basically organized into three periods: G1, the S phase, and G2, where the S phase was the point where DNA was replicated in anticipation of the upcoming or imminent cell division, where the amount of genetic material would need to be doubled so that each of the resulting daughter cells would have their fair complement of genetic material. The DNA in here is not, as I said before, visible in a typical chromosome X structure. In fact, the DNA is really the same thing. It's identical to what you find in a typical chromosome, except that it's not visible. And the reason it's not visible is that that DNA is spread out. It's very diffuse, spread out. It's in a diffuse type of an arrangement, and we call that chromatin, C-H-R-O-M-A-T-I-N. So consequently, later on, when we actually reach cell division, that chromatin will condense, and then you will see the typical chromosome structures. Chromosome structures, in the typical sense, are only visible when a cell is dividing, not during interphase, as you see in this picture over here. Okay, so having said that, let me see if I can get out of this. And what I'll do is, there we go. Okay, the very next phase of mitosis is prophase. And in prophase, you have a condensation or a compacting of the DNA material. And what happens is that chromatin material that was diffused before starts to condense, and the nuclear membrane begins to disappear. And as a result, we can look over here at a prophase arrangement. Okay, and once again, I'm going to go back and see if I can actually type in the word prophase. Let's go over here. There we go. Okay, if you look at this particular cell, and by the way, the markings of this cell, the actual delineation of where it ends and where it begins, I can show you right here. Let me go and do one of these. Now, one of the reasons why students like plant cells as opposed to animal cells in their initial studies of mitosis is that the delineation, the sharp boundaries from one cell to another are most apparent. Don't forget that plant cells don't just have a cell membrane. They also have another from that, known as the cell wall, which imparts a certain rigidity to plant cells. Yes, I know it's an anatomy and physiology class and we're not going into plants, but this is a plant. This is plant tissue that you're looking at right now. So when you look at the animal tissue from mitosis in the slide box, the delineation, the demarcation, the boundaries of these cells may not be as apparent as you see so nicely here. Anyway, my point here is that this area is a nucleus, and you can look very closely. It's starting to break down. And that purple area in the middle is the beginning of the condensation of the chromatin, where you will eventually see the chromosomes appear. You don't see that in these other cells down here, do you? Okay, so eventually the chromosomes will appear, and they will obviously be duplicated. They're duplicated why? They're duplicated because in the S phase, prior to mitosis, the DNA was duplicated in anticipation of an upcoming cell division. Why did it have to be duplicated? Eventually you're going to find out one cell is going to become two. So if you just split it in half, you would only get half DNA in one cell and half in the other. So they must be duplicated so that you get a full complement of DNA in each cell. Okay, so this then over here is prophase, and what I'm going to do is see if I can type that one in. So at the end you have something to print out. There we go. Okay, the next phase after prophase, and again, this is so undetailed. I was just trying to see whether or not I can deliver this to you with additional material. I'll probably put some PowerPoint slides in between, etc. If you look over here, let me see if I can go back and do this the way that, there we go. If you look at this point over here, the chromosomes are actually lining up on what is referred to as the metaphase plate. Now, the metaphase plate may not be apparent to you right here, but in fact, the metaphase plate runs somewhere down about so. And along that line that I just drew, you have the centromeres. The centromeres of the individual chromosomes lined up along that area. Eventually those centromeres, oh, by the way, this is metaphase, if you recall from our pre-laboratory talk. So I'm going to write that in over here. Okay, this is metaphase. Okay. And eventually those centromeres will divide. Well, not really divide, but they'll split. So that one half of the chromosomal material will go one way to a potential cell, and the others will go the other way to the poles of the cell. You can see that, let's see, where can you see that best here? Actually, it's right nearby. If you look over here, you can see the chromosomes moving to the opposite poles. Yes, I know this isn't a movie, you're not watching them move, but you can see that the original chromosome mass on the metaphase plate has now, due to the help of spindle fibers, which are not terribly visible over here, I admit, due to the spindle fibers pulling and assisting in the segregation or the separating apart after that centromere has split, we have movement to the opposite poles. This is referred to as anaphase. I guess I'll center it over there. Okay. So after anaphase, we have the beginning of telophase. And in telophase, the distinctive characteristic of a plant cell, as we said, is a appearance or the formation, not of furrowing, not of invagination. That can't happen in plant cells. The cell wall is just too rigid to allow that to occur. However, you have the formation of what's referred to as a cell plate. And if I do this over here, you might see some cells over here. I can tell that they're in telophase, they just finished telophase, because first of all, the cells are small. These cells are much larger over here. These cells are larger, and these are small. Here's the cell plate that's starting to form, or has formed rather, between the two. I should point out, let me just type in the fact that these are telophase. Let's see if I can type that in. This is great. Okay, so if you look around here, you get a general scheme of the variety of what's happening. You can see one of the things you glean from all of this is that you don't have the whole tissue moving in synchrony. You look at different areas and they are progressing at different rates. Some are slightly ahead, some are slightly behind. Some, as we said during interphase, are not even dividing at all. I just want to try one other tool over here to see how it works. So when I talk about the interface, I'll point to over here. And that works. That's a nice little tool. Okay. All right, so let's forget about that for now. So you get an idea of the distribution of the various phases in one tissue. Remember, the chromosomes are duplicated prior to mitosis. An interphase at the very beginning of interphase, which was preceded by what? Preceded by the late telophase. The chromosomes are unduplicated. When the S phase is activated or occurs, we have the synthesis or the duplicating of DNA, and just prior to the actual mitosis, we had the G2 as we explained in class. I think that's probably about enough for right now. Again, I'm not even listening to what I'm saying, so please don't hold me to anything at this point. What I'm really doing here is trying to see if some of the pictures that we have collected from the technology training grant can be delivered in a way where an instructor can talk about it. And again, we can pretend that we're sitting at your house looking down a microscope tube or an eyepiece or an ocular and seeing things together. So please forgive me if I said anything incorrectly. I'll probably find out whether I did or not after I replay it. But my main purpose right now is to see whether or not this image is clear. Do you understand me? Is the typing clear? Does this file come up without having to wait for 20 minutes? It's based on a protocol that I was able to use in the cadaver dissection movies, which is an ongoing project and should be concluded by June. In any event, please let me know. Let me know if this works on your computer. And I'm going to get out of here right now and take a look on my computer and see whether or not it comes across well. Okay, so I'm going to say goodbye for now.